Okay, now in this session we are going to discuss here the another layer of the eyeball after the outer fibrous coat, outer fibrous tunica that consists of posterior 5/6 opaque or the white of the eyeball, that is the sclera, and the anterior 1/6 that was the transparent of the eyeball, that is called cornea, corneoscleral junction of the limbus, scleral spur, and to some extent you see that the canal of Schlemm, anterior chamber, some introduction about that to cornea, 5 layers, lamina cribrosa, the weakest point on the back pole, or the posterior pole of the eyeball. All these things we have actually discussed. Now we are discussing here the intermediate of the vascular pigmented coat, also this is called. Now in this vascular pigmented coat, if you remember, we told you last time that you can divide the eyeball into 6 portions here. See, 6 portions means you can divide this like this. Six parts. Now in these 6 parts, we told you already the fibrous coat, 6 parts, 1/6 anterior, it was cornea, and after the cornea you have seen this 5/6, it was sclera, and after this is sclera. But inside, if you are going into the second layer, in the second layer where you see in this 5/6 where the sclera was there, we got this line also, and this line gave you idea that this part of the sclera is the visible sclera, which you can see from outside, and the remaining portion was the hidden sclera. This was the hidden sclera. And as for example, you see hidden sclera and visible sclera, means you can say 4/6 is the hidden sclera, 1/6 is the visible sclera, and remaining 1/6 you know that is cornea. Now as far as your 4/6 are concerned, under that whatever layer was actually lying, the second layer, the part of the second layer, this one, which was hidden by the hidden sclera, this is called the choroid. Choroid only, see the vascular pigmented coat, which I am actually telling you, consists of 3 distinct regions here. The one region which in fact consists of just posterior 4/6, that portion which is overlaid by the hidden sclera. But the next 1/6 which is overlapped by this, means visible sclera, this portion of the vascular pigmented or the next coat, under that is the ciliary body. This is the ciliary body. And after that comes the next 1/6, that is called the cornea, and under the cornea will be lying here the third thing, or the third part of the vascular pigmented coat, that is called the iris. And you know that iris is not full, complete. It has got the deficiency in the middle, that is called the pupil. That means 1/6, the last 1/6 will be comprised of iris and the pupil, you can say. Pupil is something which is a deficient thing, it is a hole in the middle of the iris here. So this will be something, these are called here, like, and in all these 3 segments together is called the uvea, also, or called the uveal tract. So much so that the iris and a part of, you say, most anterior part of the ciliary body together is called the anterior uveal tract, and the rest of the ciliary body and the whole choroid together is called the posterior uveal tract. So call it the anterior and posterior uveal tracts also. So in this way we are going to discuss various parts of the uveal tract here. Now you see, first thing is obviously the choroid, which you know that it is forming the posterior 4/6 of the uveal tract. would constitute a major part of the posterior uveal tract, and overlying this part of the uveal tract is the hidden sclera. You are watching this hidden sclera, 4/6, and under that you are seeing this green color. This green color under this will be the choroid. It is roughly 4/6. I am telling you. This is choroid, posterior 4/6 of the middle coat. Again, you are watching this green color, and we already know that then the middle 1/6 was, you see, after you see the posterior 4/6, it was choroid, and then the middle 1/6 will be this green color, that is the ciliary body, and the last 1/6 which we will telling you is the iris. It is not 1/6 full, because in the middle there is the hole. This is called the pupil. So, it is not full 1/6 because there is a deficiency in the middle, and this deficiency can be adjusted, can be enlarged, can be narrowed down. Narrowed means there will be meiosis, or enlarged it will be mydriasis. Mydriasis means dilatation of the pupil, means enlarging its diameter. So, ciliary body is there, and the last will be, you know, that it is called the iris, and in the middle we have a hole. Iris is not a half circle, it is a full circle, obviously, because we are cutting here the half of the eyeball. That is why it looks one half, you see. So, iris was there, but look at the choroid now. Choroid, you see already, this anteriorly, you know that it merges with the ciliary body. Already I told you that it merges with the ciliary body, and anteriorly it merges with the ciliary body, and together I told you that, and ciliary body anterior merges with the iris. So, anteriorly it merges in the ciliary body, and both forms the posterior uveal tract, but we told you that iris forms the anterior uveal tract, iris and part of the ciliary body also. And posteriorly, when you will go posteriorly, like ciliary, like your sclera, this is also perforated by the fibers of the optic nerve, to which it is firmly attached also. So, choroid is actually posteriorly attached here with the fibers of the optic nerve. So, it is perforated also, just like perforation of your sclera was there in the, and there you call them the lamina cribrosa in case of sclera, the weakest point you see that of the sclera. And if you are looking at this inner surface, already I told you that inner surface is firmly attached to the, you see, if for example you see inside is because it is highly vascular and pigmented coat, and highly vascular and pigmented coat, if you are actually seeing this is highly vascular and pigmented coat here, this one here is a lot of blood vessels, network of blood vessels is there. This is called the choroid plexus, and inside it is because cornea, as you say, retina has the 10 layers. So, outermost layer, which is called the epithelial layer, which is firmly adherent with the inner surface of the choroid here, but the outer surface of the choroid is in contact with the sclera, 4/6 sclera, hidden sclera, though the total sclera is 5/6. So, it is hidden sclera, under that it is actually passing. So, its inner surface and outer surface you should have an idea, and you see this here, this is the plexus which is in the choroid here, and so much so that under surface you see that its inner surface is adherent to, firmly adherent with the outermost, or the layer number 1 of you can say layer number 1 of the retina here. That is why it is actually giving blood supply through diffusion to the outer 1/3 of the retina, outer 1/3 of the retina, including rods and cones. They depend for their blood supply, for their oxygen and other thing, and the nutrition on the choroid, through the choroid plexus. And you see this choroid plexus has got the blood vessels, the ciliary blood vessels, others enter and form a huge plexus here, which not only supplies the underlying 1/3, or you see outer 1/3 of the retina, which is adherent to its inner surface, but the thing which is adherent to its outer surface, that is called the sclera. How much? The 4/6 of the sclera, which is avascular also, is also depending on the choroid plexus for its nutrition and other things here, and its metabolites and drainage here. What will happen with the rest of the 1/6 sclera hidden? This is called the visible sclera, because it was under the cover of the conjunctiva. So, it will be supplied here by the conjunctival vessels. That means every time, and you see that. What about the cornea? It does not have the conjunctiva, because conjunctiva becomes continuous with its epithelium. Okay, we do not have any problem, because it does not need the blood supply, because deeper to that or posterior to the cornea is the anterior chamber, and anterior chamber contains the aqueous humor, and nature has actually caused the blood supply to disappear from here, because blood is opaque, and nature has to keep this compartment in a transparent environment. That is why nature has provided here the aqueous humor, and aqueous humor is doing almost all those things which the blood is expected to do. So, you see this vein, and veins are also leaving here, and when few big veins are leaving here, the surface of the choroid penetrate the sclera to go to the systemic circulation, and these are called the venae vaticosi. These are 4 or 5 veins, 2 have been shown here. This is called the venae vaticosi. And choroid, this is called the choroid. The choroid plexus has been shown here. The ciliary vessels are actually, ciliary arteries are entering, anterior ciliary, posterior ciliary arteries forming the plexus all around. At the same time, veins are leaving also. These are 5 to 6 or 4 to 5 venae vaticosi are leaving to enter into the ophthalmic veins, and you see that will drain into the sinuses and other systemic circulation. We told you last time that the optic nerve is surrounded here by the meninges, because it is basically the extension coming from the brain. So, it is a this sclera is becoming continuous. You see, sclera is a, because when in the eyeball it was sclera, name sclera, the same material around the optic nerve it was a durameter. Is mesenchymal, you see that again. Mesenchyme here is forming not the sclera around, because material is same, but you see is a fibrous tissue, material is same, but around the eyeball its name will become sclera. When around your optic nerve, when its name was a arachnoid mater, then it was a pia mater, 2 layers here again on this section. Both of these layers in between, they have got the subarachnoid space. I am making dots in them. The subarachnoid space contains the CSF. But both these maters, you see that arachnoid as well as the pia mater both fuse to become continuous with obviously the second layer, that is called the choroid. That means in the choroid we do not have 2 layers, it is just single layer, vascular pigmented. This vascular pigmented core means your subarachnoid space stops at the back of the lamina cerebrosa, or you can say it stops at the back of the optic disc. That is why if there is intracranial pressure raised and in the subarachnoid space the pressure is more, the optic disc actually will be pushed from back. If pushed from back means there is a bulging. And if the optic disc is actually pushed from the front, because there is intraocular pressure more, there will be cupping. So, this is because of the intraocular pressure present here, because around the eyeball we do not have any subarachnoid space, though we have the continuations of the pia mater and arachnoid mater. Basically, arachnoid mater and subarachnoid and arachnoid mater and pia mater, this is one layer, and this one layer actually splits into the 2, making a space between, that is called the subarachnoid space. And here you are actually watching the choroid. So, choroid is continuous here with the pia mater and arachnoid maters without intervening subarachnoid space in it. Subarachnoid space stops at the posterior pole at the optic disc area. This was about the choroid here, and now you are bringing concentration on the ciliary body. If you look at the ciliary body, I have told you that already that the ciliary body, it occupies the middle 1/6 of this one. It is the middle 1/6 of your vascular pigmented cord or the middle cord here. You see this, and incidentally, overlying here it will be, we have given this this line, visible portion. So this is the visible portion. This sclera is the visible sclera. Means a visible sclera. This is underlying the visible sclera. Means 1/6. So this lies actually in the 1/6 here. So middle 1/6 I say, and I have already told you that if this is middle 1/6, so anterior 1/6 will be the iris, and posterior 1/6 it is continuous with the posterior 4/6 it is continuous with the choroid. So you see the middle 1/6 here, and on the back you see already the choroid here. So it is actually sandwiched between anterior 1/6 and posterior 4/6 to make the total 6 parts here. And we have again given you some idea that anteriorly it merges with the last part of the vascular pigmented cord, that is called the iris, and posteriorly with the choroid. And posteriorly it merges with the choroid, and along with the choroid, ciliary body together is called the posterior uveal tract. And anteriorly the iris is forming the anterior uveal tract. And already you see here, if you are actually looking at this, another very important thing is like the choroid inside was in contact with the retina. Retina is the outermost layer out of 10, the first layer, this is called the epithelial layer, and its outer surface of the choroid was in contact with the sclera, posterior 4/6 of the sclera. But this time when you are actually bringing this now, the ciliary body, the inner surface of the ciliary body will show you 70 to 80 certain foldings here of the epithelium. These are called ciliary processes. You see, sir, here are 4 or 5. This is in section only. On the other side, it will be forming here a total ring, that is called the ciliary ring also. So inner surface, you are watching these ciliary processes, and if the ciliary processes are there, so you can say if the ciliary processes are there. So you say if the processes here, process actually is lifted here, and then will be the groove. Then process lifted, then is the groove. So you will actually be wherever there is a groove, this groove will be filled by. Groove will be filled by the material which is present here. This time here is the material that is called the aqueous humor, and on the back side behind the lens here, I will tell you this lens, but behind this lens it will be present here the vitreous humor, you call it. Vitreous means a glass, it is just like glass. So it's called the vitreous humor. We call them vitreous, I will call them vitreous body also. So vitreous humor or the vitreous fluid, in fact this is a gel like fluid that will fill the gaps which have been created by these folds, and where the fold is penetrating into the vitreous will be displaced. Means this is reciprocal grooves on the anterior surface of the vitreous body here. So reciprocal groove means where there is a folding, dip is here inside, vitreous will fill it. When there is a folding, vitreous will leave the space. So it is making actually a grip over the vitreous also. So anteriorly it is here, and when you are actually looking at this, that these ciliary bodies or the ciliary processes which have been created here are very important, because ciliary epithelium which is actually folded here to increase the surface area, and from here is in fact secreted the aqueous humor. And ciliary processes are important from this point of view. At the same time, the ciliary processes are actually giving attachment to these fibers also. And these fibers are called the zonule. You call them zonular fibers, or its name is suspensory ligament of the lens, because it is catching hold of the lens here, because they are holding the lens, not from one side and other side, from all sides. Total circumference of this biconvex crystalline lens, they are held by from all sides by these fibers here. We will show you that in the lens also. So ciliary process is a ciliary process, inner and outer relation you have again seen, and where your ciliary body finishes here, surely outside the sclera will finish, and when your ciliary body finishes, your vascular pigmented cords iris starts, and outside starts the cornea. Means here an angle will be created that should be called as the iridocorneal angle, and incidentally between the iris and the cornea, this is anterior chamber. So iridocorneal angle can be called as, the other name will be the angle of the anterior chamber, and this angle of the anterior chamber is filled here by the trabecular meshwork, and this trabecular meshwork will finally drain into the canal. This is called the scleral canal, that is called the canal of Schilem. So you see this is the canal of Schilem here, and here is the trabecular meshwork, and this is the angle, this is called the iridocorneal angle. Why iridocorneal? Because here the cornea finishes in the second layer, and immediately you see that here here the cornea finishes in the outer layer, and here incidentally the iris finishes also, where both will meet here. Meet means angle is created, and this angle, yes, it is. The other side also. Other side, there are not two sides, they are total circle. After that, you watch here the ciliary process. I told you that ciliary process is there. So, the ciliary processes you have actually seen here, the ciliary process, but inside the ciliary process is deeper, you have got the muscle here. That muscle takes origin from the scleral spur, which I have told you that just behind the canal of Schilling. So, here these are the muscles here, the ciliary muscle, which consists of, which is smooth muscle, whose function is to focus the lens. Actually, to when they will contract, the traction you see on the ciliary and the zonular fibers will be less, means they will be relaxed, means when the ciliary muscle contracts, zonular fibers they relax, and when zonular fibers will relax, the elastic capsule, you see that elastic lens, in fact it will bulge more, because zonular fibers are getting relaxed when ciliary muscle contracts, because the ciliary muscle, it's a rounded way, in a sphincteric way it is contracting. So, this will be bulge, and bulge means its refractory power will increase. Refractory power increases means it will focus the, means it will refract the rays earlier, means they will actually refracting them more, means refractory power increases here. Means you will focus those objects which are being brought here near to your eyeball. This is actually called the accommodation. And when you are actually drawing the object away, these muscles will become relaxed. If they become relaxed, the zonular fibers become tight. If the zonular fibers become tight, the lens will become flatter. Flatter, this focal length will decrease, or you say focusing power or accommodation power or refractory power will decrease. This is something accommodation, because one third of the refractory power lies with the lens here, and that refractory power is accommodating. This is not fixed. 2/3 is being actually given here to just to the cornea. We will tell you that in refractory media also. So, here this is the ciliary muscle, whose function is obviously which is origin is from the scleral spur, and there is a insertion here is to the ciliary processes. So, it is actually moving the ciliary processes here, and to the ciliary process you know that it is attached, see it is attached here to the ciliary processes, and to the ciliary processes attached here. So, indirectly zonular fibers are being affected here, and obviously not zonular fibers, basically the lens is being affected here. You see this is ciliary processes and zonular fibers. So, this is the muscle which is in fact is affecting the ciliary processes, then to the zonular fibers, and obviously to the lens here. One of the most active muscles in your eyeball, probably in the body, is the ciliary muscle here. A ciliary muscle, I told you that fibers relaxed, suspensory ligament become taut, lens will become thinner when they are relaxed. But when the ciliary muscle are contracted, suspensory ligaments will become relaxed, and they become relaxed, tension on the lens is less, elastic lens will bulge and will focus, having refractory power more, has the capability to focus the near objects here. Ciliary process, already I have told you that 70 to 80 small ciliary processes. So, watching the ciliary processes, few of them have been shown here, and whatever space is there inside, you see choroid will fill them, and whatever foldings they will push, actually they will push into the choroid will be displaced. So, they are interdigitating, and from the ciliary processes will emerge the zonular fibers. We told you that the ciliary body which you are seeing here is not just here, and is not just on the other side, it will be actually throughout. So, it is forming here a ring that is called the ciliary ring. So, that means the ciliary ring is there. If the ciliary ring will be there all around, this is ciliary body. You see in cut section it will look like this. On this side it is here, and this, and one side here you are watching here, other side is watching here. Otherwise it is like ring, and from the ring will emerge what? From the ring ciliary processes. So here will be the ciliary processes, medially will be the ciliary processes. From ciliary processes will emerge the zonular fibers from all sides, and there is a gap, and this gap will be filled by the lens. So if you are anterior, means if you are here in this chamber, you see, okay, you say here in this chamber, this is called the anterior segment of the eye, in which there will be iris in the middle, diaphragm in the middle, which will make the anterior segment into the anterior chamber and posterior chamber, but this is called the anterior segment here. And behind the zonular fibers and the lens, whatever behind you will see, this is called the posterior segment of the eyeball, which is actually the filled by the vitreous body. So anterior segment of the eye and posterior segment of the eye. So there are two segments also separated here by the ciliary ring, as such you can say, ciliary ring partly and partly by lens and zonular fibers. So ciliary ring has been created by the ciliary body along with the ciliary process, etc. After this, you see the third thing among the among your uveal tract. In your uveal tract, the third thing is the iris, which you know that is under the cornea, 1/6. Obviously, cornea is 1/6, and iris is not total 1/6, because there is a deficiency also. So deficiency and iris together is 1/6, and the deficiency is lying in the center. And the center means if you pass something, is like a hole, it's a dark hole, it's a black hole. Through the black hole, why is it black? We will tell you why it's a black. Black because all the rays which have actually entered, light rays which have entered, they are all the photons, they are absorbed by the walls of either directly or indirectly, they are absorbed by the walls of the eyeball, especially by the choroid, which is the vascular pigmented coat, contains melanin. And if it is actually, remember, you see, if they have been absorbed here, and whatever is there, this will be absorbed here by the pigmented layer. This is called the epithelial layer of the retina also. But nothing returns back. When nothing returns back, the hole through which it has entered, the light rays which have entered through the hole, that hole will look dark color, it will look black, because nothing is actually reflecting. If something will reflect here, like happens where there is a melanin deficiency, like in albinos, you see that this iris will look shiny, it will look now, you see this is having some light in that, it will not look dark or will not look black. So photophobia will be there. So you see, this will be actually the iris and the pupil here. So anteriorly you are watching here the iris, and iris is made up of, I will tell you, this is made up of the two muscles here. It's not just muscle, it is having a pigmented layer, non-pigmented layer, muscles, etc. So iris occupies 1/6 along with the pupil, not just pupil. You see that it can be even the pupil dilates here. Pupil dilates, half will be filled by the pupil and half will be filled by the iris here, and the pupil contracts. So surely less than 1/4, even less than 1/4 will be occupied by pupil, and 3/4 will be occupied by the iris. This depends. So it will be aperture, that aperture pupil will be regulated by the contraction and relaxation of the muscles. And we told you again, in this layer we have seen anterior 1/6, this was a iris, the middle 1/6, it was again green color, this is a ciliary body, and the last 1/6 4/6 it was the choroid, to make them all 6 parts here. And relation will be posterior 4/6, overlying it will be the hidden sclera. Next 1/6, you know that is a hidden overlapped by visible sclera, and the first 1/6 that was hidden by, this time not hidden as such because you can see them, and the pupil is also there. The size of the pupil which you actually see from outside is not the actual size, because it is magnified by, because you are seeing this across the cornea. So if you actually remove the cornea, the pupil will not look the same size here, because the cornea is already there and you are seeing it through the cornea, so it is magnified, and when it is magnified, so actually you are seeing the pupil. So pupil is not having the same diameter, real diameter you are not actually seeing from outside here, because you are seeing it in a magnified way. And if you remove the iris, for example, if you remove the cornea, then you will see this hole in the middle of the iris, that is called the pupil, in its original diameter here. So watch it, the diameter of your pupil that you are seeing, basically it is magnified, it's not real. Okay, ciliary body and choroid, you have seen this also, and pupil, you have discussed the pupil. And we have told you already that when you are actually drawing this line here across the lens, so anterior to this line will be the visible part of sclera. I say visible part of your eye, that will be hardly, you say, 1/3, and posteriorly it will be 2/3. This posterior 2/3 where there is vitreous humor, this is called the posterior segment of the eyeball, and anteriorly where there is aqueous humor, that is called the anterior segment of the eyeball. And the anterior segment of the eyeball is in fact partitioned by the iris also. Make the anterior segment into two chambers, one between larger one between cornea and the front of the iris, this is called the anterior chamber, AC, and behind your iris up to the lens comes here, this chamber is called PC, that is called the posterior chamber. Means anterior segment of the eyeball is divided again into anterior chambers and posterior chambers. And if you will actually look at iris, inner surface of the iris, you can see this inner surface of the iris, this is the endothelium of the iris, you see, sorry, inner surface of the cornea, that is the endothelium of the cornea, which is forming the anterior wall of the of the anterior chamber. And outer surface, it forms cornea. So this will be here, this anterior surface which you are seeing of the iris forming the posterior wall of the anterior chamber. This is, so outer surface is forming the posterior wall of the anterior chamber, whose anterior wall is formed here by the endothelium of the cornea. And surely the posterior wall is not just formed here by the anterior surface of the iris, but also pupil also, because they are deficient. But the posterior chamber which you see here, so the inner surface of the iris is forming the anterior wall of the posterior chamber, where the posterior wall of the posterior chamber is made here by the anterior surface of the lens. You see in this also, if the iris outer surface, you see outer surface of the iris. Here is your AC, anterior chamber. So this outer surface of the iris is forming the posterior wall of the anterior chamber, where the anterior wall of the anterior chamber, already you know that is formed by the cornea. Cornea is the endothelium. But when you are seeing the posterior layer as the posterior surface of your iris, this is forming the anterior wall of the posterior chamber, where the posterior wall of the posterior chamber is formed by the lens. So that means anterior chamber and posterior chamber, they are communicating via the pupil, and the fluid which is being produced here by the ciliary body first should come into the posterior chamber, and from posterior chamber it can pass here through the pupil to come into the anterior chamber, and from anterior chamber it will accumulate here in the angle of anterior chamber, which are filled by the trabecular meshwork, which will suck them, because inside them is the capillary network, and capillary network has a negative pressure, so they will suck them and they will put them into the canal of Schilling, which you know that is lying in the most anterior part of your sclera, and they will drain them into the systemic circulation, just like your lymph. It is not lymph, it is not blood, but it is performing the functions of both, because we have to make this area totally transparent, because the rays which will actually strike the cornea will be refracting and will pass through another transparent fluid here, another transparent environment, that is the aqueous humor, and then it will strike the opaque environment, that is called the iris. No problem, iris will give the deficiency in the middle through the pupil, they can enter through the pupil, and they will come into the posterior chamber, and posterior chamber again same aqueous humor, again transparent environment. When you are actually striking now the lens itself is crystalline, transparent, and when you enter into the vitreous itself is gel like transparent body here. That means you can see your retina direct without obstruction. And after having some physical changes here, that means refraction, everything, so that it should focus now on the retina. And already we gave you idea that there is the angle of anterior chamber also. Angle of anterior chamber is present here. It forms the angle of anterior chamber. Iris forms the angle of anterior chamber. Why? Because outside the cornea is also finishing, so iridocorneal angle will be created, whose name is angle of anterior chamber. Pupil, already we told you, perforated centrally, the iris perforated centrally, means deficient basically. It's not perforated as such, but it's deficient centrally. Pupil is very important gate from the posterior chamber to anterior chamber, allowing the aqueous humor. Drainage of aqueous humor is here, and through that obviously is not just a drainage of aqueous humor, but the passage of the light, rays from outside to in up to the retina. Surely the gateway will be the pupil, and you have to regulate the pupil according to the need of you see this is here, this will be the pupil and the rest will be the iris. And here you see this pupil, almost half will be the pupil, more than half, and this less than half will be the muscle of the iris here. Here less, here still less. When the bright light will come, you will actually have this probe. When the normal light, this. When the dim light, you see this. Means it will be maximum contraction of you can say of the radial muscles, we call the longitudinal muscles, because they have the longitudinal muscles, larger one, and you have got the circular muscles. Circular muscles will be surrounding the pupil, and then surrounding the circular muscles will be the longitudinal muscles. The longitudinal muscles, when they contract, the pupil will dilate. Means the longitudinal muscles should be supplied here by, because will dilate means more light will enter. Means this is emergency. Means this muscle should be supplied here by the nervous system of emergency. Means of the sympathetic system. Means this muscle which is longitudinal, we call them dilator pupillae. They are under the control of the sympathetic innervation. While those muscles which are actually surrounding the pupil, they are circular. We call them constrictor pupillae or sphincter pupillae. And these muscles, when they contract, the pupil will become narrowed down. Meiosis will take place. When these muscles are contracted sympathetic, so they are supplied here. The circular muscles, sphincter pupillae, they are supplied by the parasympathetic. When they contract, the pupil will become narrowed down. Meiosis will take place and less light will enter. This is you are out of emergency, you are at rest. So parasympathetic is working. The sympathetic and the parasympathetic. You see, these radial muscles, you can say dilator pupillae by sympathetic. And sympathetic means when they will reach here, there must be postganglionic sympathetic fibers, and their cell body is lying in the superior cervical ganglia. That means if the superior cervical ganglia is compressed or pressurized here, you will see that these muscles will become relaxed. When they become relaxed, narrowness of the pupil will take place. Dilatation will cease to occur. When this dilatation will cease to occur, narrowness will take place, because parasympathetic will be dominating. This is called the meiosis. That happens in Horner syndrome also. But as far as your constrictor pupillae is concerned, or you say sphincter pupillae is concerned, this is under the control of the parasympathetic. Parasympathetic, obviously the postganglionic parasympathetic fibers will reach here. Postganglionic parasympathetic fibers, why? Because outside the eyeball is lying here a ganglion, that is called the ciliary ganglion. Into the ciliary ganglion will come the periganglionic, but from ciliary ganglion will leave the fibers which will actually enter into the sclera to find the constrictor pupillae fibers. So they will be postganglionic. Short nerves will be there. These are called short ciliary nerves. But the periganglionic fibers are something different here. They come along with mostly you see that it will come, parasympathetic fibers come along with the oculomotor nerve. Oculomotor nerve contains more than 95% fibers voluntary, but they have on their shoulders the responsibility of carrying at least 5% of their own load, which should actually be supplying this constrictor pupillae. Not just constrictor pupillae, but inside is lying here another circular muscle also, whose name is ciliary body. So they will be supplying ciliary muscle also, ciliary muscle of the ciliary body. As autonomic innervation, I have told you, and you are watching these muscles here. These muscle fibers are dilator pupillae fibers from all sides, and these are the constrictor pupillae, or say sphincter pupillae also. and between the gap which has been left here, this is called the pupil, this is called the pupil, and behind you know that is the lens. These are the zonular fibers and this is ciliary body, and you are watching here canal of Schilm, and this is the trabecular meshwork in the angle of anterior chamber. And you see from here in front, this is the visible part of sclera, and you are watching here the cornea is overlying the iris. The color of this blue color which you are actually seeing is not that of cornea, this is actually of posteriorly placed iris. This is the pigmented layer of the iris, because iris belongs to the vascular pigmented coat, the most anterior 1/6 of the vascular pigmented coat, with the deficiency in the middle, that is dark. It is not dark, it is deficient. It is looking dark because the rays are entering inside them but are not reflecting. So, see, this is radial fibers here, and these are circular fibers. See these cut section, see these radial fibers, and they are dotted here, means they are cut, they have been cut here, that they are here. So, this, see, this is cut section and their cut section. These are longitudinal fibers, dilator pupilli. When they contract, they shorten. Shorten means the pupil size will increase. When they contract, the pupil size will decrease. Now, after this was something about these three layers, which were the three segments of the middle layer. Now, watch some about the refractory media. First, do you see that first refractory medium, the transparent front of the eye that is covering it from outside, it is called the cornea. It refracts it when the light strikes here from outer environment, that is the air, and it strikes here. So, the difference between the density of your specific gravity, also the density of your lighter medium, that is the air, and the this denser medium, that is the cornea, the maximum difference is here. That is why the refractive index of this medium is most. So, it refracts the light so that it enters into the eye, it refracts it most. Why? Because the rays which are parallel coming from here, they should actually refract and will pass through the pupil, because pupil size is not very big. So, you have to, parallel fibers cannot enter through the iris here, parallel fibers will enter through the central part of the iris. So, they should be refracted most. That is why the cornea is actually having the almost 2/3 of your total refractory power is with the iris here. This is around, you can say, 43 diopters, and this 43 diopters, there is a refractory power that is fixed always, is not accommodating at all. It will not become 42, 41 or it will become 45 or something. No, it is fixed. So, cornea is the first refractory medium here. After that, you enter into the anterior chamber, where there is aqueous, transparent watery fluid it is, with a very low protein concentration. You already know it is being secreted here by the ciliary processes, ciliary epithelium, because processes are the foldings of epithelium, not of the muscles. So, it is here, and it comes into the posterior chamber, and after the posterior chamber, it will actually leave through the pupil and will come into the anterior chamber, and in the angle of anterior chamber, it will be sucked. You see that it will be absorbed by the trabecular meshwork, so that it should go into the canal of Schilm. From canal of Schilm, it will go to the systemic circulation. So, the composition you have seen here, and then the posterior chamber, you are actually, posterior chamber is a very narrow or potentially say space here between the back of the iris and the zonular fibers here, that is all. Not between the iris and the lens, because the lens is actually being touched, the anterior surface of the lens is being touched here by the iris, does not have enough space here. And aqueous humor, you know that, maintains the intraocular pressure, because inside there is not air but the fluid, which will give you the which will inflate the fluid just like balloon. So, it will remains like globular. Also, it provides nutrition. Why it provides nutrition? Because we do not have blood here, we have a avascular epithelium. Epithelium is the avascular of the lens as well as of the cornea. Though iris, we do not need the aqueous for the iris, because iris already is the vascular pigmented, but the lens and cornea, they are not, their epithelium is there. Their epithelium means you have to give the nutrition here. Plus, it will have antioxidant, vitamin C. This called the ascorbate has to be driven here into this to bring about the antioxidation. Is called here fluid. It is fluid, so it has a immunoglobulin A also, which is giving here the immunosurveillance. provides inflation for the expansion of the cornea also, because cornea is having a bulge, you see more curvature, more focusing power it will have, more refractive index it will have. Also it is giving the corneal protection, and behind is giving the protection to the lens as well, and prevents the dryness of the eyes. That's a very, very important fluid. You see that when this production is more, glaucoma will take place, or if it is the drainage is obstructed, again the glaucoma will take place. Production more, angle is open, so you call them open angle glaucoma. When the angle is narrow but the production is normal, you will call it the angle closure glaucoma. Production already I told you that from the ciliary processes, from ciliary body, but from ciliary epithelium. It will come into the posterior chamber. And drainage, we told you that again, this is the arrows depicting the drainage here. In the posterior chamber they come, then through the pupil they will come in the anterior chamber, accumulate in the anterior chamber angle, and the trabecular mesh will carry them, and into the canal of Schilm they will enter. This is almost, you can say almost 90% of drainage is like this, but the 10% drainage basically is through the uveoscleral drainage, means from here uveal tract here, so here is the sclera, so from here also to the episcleral blood vessels it will actually be going here. So that means from the choroid plexus here it will be also draining some aqueous, you will actually be draining from here in the limbus and other areas also. So here are the subconjunctival blood vessels here. So some will be through this, drainage will occur through this as well, but this is only 10%. 90% is through this internal system. This is called the trabecular meshwork. And I told you that glaucoma, intraocular pressure when it is raised here is called the glaucoma. It's not just intraocular pressure raised will be called glaucoma. Glaucoma will be when not just intraocular pressure, but it will be optic neuropathy as well as visual field defects. Then you will call it the glaucoma. And usually when there is increased resistance to the outflow at the angle of the anterior chamber in the meshwork area, this will be called the angle closure glaucoma. Otherwise you can call it the angle, what's called the wide angle glaucoma also. Aqueous humor, we have told you this drainage also. The posterior chamber you are watching here, important thing is only posterior chamber is a narrow space behind the peripheral part of here, not the central part. I told you the central part is just in touch with this interior surface. So we don't have it. This is only small area, you said. This small area is just to receive the aqueous humor, because this is the area where the aqueous humor drop by drop is being received. This area. On this side and this side, not in this and this, it's totally surrounding. People already we discussed. Why it is black and others we say. In people, especially you see that people in human being, it is a round area, like it is round. But in cats it is a vertical slit, and in goats it is a horizontal. So different interesting facts about these people. Angle of anterior chamber, we have already told you that. So depth of the angle of anterior chamber, so depth of the anterior chamber total is 3 mm. But if it is less than 1 mm, because it decreases with the age, if it is less than 2.5 mm, then obstruction to the flow can take place here. This is called the narrow angle. Then the angle will become narrow also, because when it will become here it is in normal, when it become now like this, you see angle will become narrowed. Previously it was wider. So narrow angle glaucoma might take place in old age. Angle already we told you that trabecular meshwork there, and aqueous humor is drained. Canal of Schilm, we told you that scleral canal connected to the anterior chamber angle through the meshwork here, through the scleral veins here. Trabeculae, we told you this meshwork is an area located in the anterior chamber which is responsible for draining the aqueous humor which has reached here, and then put them into the canal of Schilm. It's called the trabecular meshwork. After that you see the lens. Lens is composed of, you see again is the biconvex. It's a biconvex lens, flexible tissue, held from all sides by the zonular fibers here, refract the along with the cornea, it will refract the light here on the retina. Here it actually carries only 1/3 of the total refractory power, but that 1/3 is adjustable. Cornea was doing 43 and he is doing actually 18, but this 18 is important in a sense that it is adjustable according to the object which is either near to your eye, where you need more focusing power, and it's away here, and this adjustment is done here by the ciliary body that we discussed previously. And if you are looking at this, the lens capsule is a smooth transparent basement membrane. Basically this red color which you are actually seeing here, this is the outer surface, this is the capsule. This is basically basement membrane, and inside you will lie here, this one here, this is the epithelium. Epithelium is lying deeper to the capsule. So capsule will actually represent it will represent the basement membrane. So lens is a smooth transparent basement membrane that completely surrounds the lens. The capsule is elastic, you know that, composed of collagen fibers, but the lens epithelium is only located in the anterior part of the lens, not on the back side. You see, back side we do not have epithelium. Anterior side, deeper to the capsule here. Almost you see all this is simple cuboidal epithelium. So means you have got the capsule and now you have got simple cuboidal epithelium. Deeper to that will be this, and then will be the lens fibers. These are called the lens fibers. These are long, thin, transparent cells which are firmly packed, you can say. You see this lens fibers. You see, this is epithelium, this is capsule, and underlying will be the epithelium, and then are the lens fibers, but above the collagen again. You see the lens here. You see the iris touching, and in between the pupil. See this lens there, and you are watching here the zonular fibers. You are seeing from inside here, so that the zonular fibers from all sides. This will give you look that how your lens is in fact located, and how the lens from all sides is being held by the zonular fibers. And here will be the ciliary processes here. Lens, you know that is the focal length changes according to the ciliary body action or ciliary muscle action, and accommodation will be carried out. Lens is more flat, and I have told you that anterior surface, you see a more flat in anterior side, it is flatter, you see it is on the anterior side. The lens is more flat on its anterior side, like it will be flat on the anterior side, and posterior it is bulging like here into the choroid. It is, I have told you that 18 diopters was its refractory power, but that is adjusting, accommodating. These are the zonular fibers which will be actually holding them from all sides here, which called the suspensory ligament also, which are held by the ciliary body. Zonular fibers, this green color. After that we watch the vitreous, which is behind the lens, means under the choroid, you see, means in the posterior ones, posterior 4/6, means in the hidden part of a eyeball. So it is a gel like substance here, which is in fact putting pressure here on the retina here, because in touch with the retina. It is a jelly like mass which occupies the posterior 4/5, you can say, or you can say posterior 4/6, you can say, almost here, but it is a little bit anterior also, but you see up to here, means it is actually supporting the lens also. The lens will bulge behind if choroid degeneration will, vitreous degeneration will take place. You see this whole green color, it is, means it is touching your even iris, ciliary body also, but not the full. So you can say posterior 4/6, and to some extent even this middle 1/6 also, but less than 1/6. You can say okay 4/5 of the total eyeball, but you can say okay 4/6 also, but you can say 4/6 okay generally, but it's more than 4/6. And 99% is water. It's made up of water. Very little cellular and fibrous contents will be there, but its consistency is very, very important. The reason is that because when this vitreous degeneration will take place, you see vitreous degeneration will take place, it will contract. When it will contract, so eyeball size will decrease. Not only eyeball size will decrease, the pressure on the retina's two layer will be less also. I will tell you that retinal detachment will take place as a result of vitreous degeneration. You see vitreous body, green color. It fills the space between, you know, that lens and retina. You see lens and retina, all this space has been filled here. So, putting pressure on the lens anteriorly, so the lens should be stabilized, and putting pressure from all sides to the retina, so that the retina should be actually in contact with two layers also. Now, if you see this vitreous all around, this is vitreous body. Now, this vitreous only if you are totally vitreous, you will see that near the center you see pale, so pale color you are looking, because here it is more fluid, but in the cortical zone you see that in the periphery it is more gel like, in the center is more softer, and is surrounded here by the collagen, because here it will be surrounded here by here see where it is in touch with the retina, it is having a thin membrane here, you can say, like made up of the collagen here. This is called the vitreous membrane, now you call them hyaloid membrane also, separating it actually from the rest of the eye. Looks like it is like a body, you can bring actually it's outside, but at multiple areas it is in touch, it is in contact. Collagen fibrils, which are actually present here in the periphery, they attach with here, they attach with the optic nerve disc here, where the disc is here, so they are attached here also. So that you cannot actually detach the entire, you say you cannot bring this body outside here, because it is attached here. It is attached where your retina finishes here, I will tell you later, but where your retina finishes there is the ora serrata, this area is called the ora serrata, fixed here also, like tent you see that. And even on the back side you see that on this side lens capsule I have told you also, so this will actually fixing with the lens capsule as well. So these are some attachments here, so that if you want to bring this whole body outside, you will actually be not allowed by the attachment of this choroid with the underlying optic disc or underlying ora serrata or underlying back side of the capsule of the lens here. They will not allow you to go away. You have to actually detach them, then the ciliary body will be outside in your hand, the vitreous body will be outside your hand. So these attachments are important in this sense. So this was your class. In the next we will be actually discussing here very important layer, that is called the inner, the real GAMP layer is coming now. All these were actually the decoration of that last layer which you are going to discuss here. It's a very important structure, that is called the retina, and with the retina we will be dealing with some discussion about the retina's continuation, where the impulses will be actually traveling back here to the thalamus. This is called the optic nerve here. This will be your next class, inshallah.